Hey guys, here's your new video. This one's on genetics. In the first part of genetics, we're going to call Mendelian genetics because they were named after Gregor Mendel. These are simple genetics where a couple or three rules really apply. So this unit's primarily going to be just about us working genetic crosses, the genetic problems. But there's some important vocabulary that you do need to get down. So let's get into it. First of all, don't write this slide, just simply read this slide. It's very important. I think it's very good information too to figure out who Gregor Mendel was, and I'll add to the story a little bit in class next time I see you. So here are those terms. This is part of the important vocabulary that you and I need to have. I mean, I've already got it. In order for you to understand what I'm talking about, I'm going to use the word gene and allele, which sometimes seem similar, but they're not. I'm going to use the word genotype and phenotype, which kind of sound a lot alike, but they're not. I'm going to use dominant recessive, and then less, I'm going to use law of segregation, law of independent assortment. We'll use Punnett square a whole lot. We'll use the word monohypercross and dihypercross some, as well as the word filial, because it actually is the word that stands for the F in the F1 generation. So write all these down, start getting familiar with them, at least saying them out loud. So Mendelian genetics is based upon three laws. Mendel discovered this as he was working with his pea plants. Law of segregation simply says every gene has two possible outcomes, two alleles, two possible outcomes, and in one organism, both of those alleles cannot go into the making of the new organism. So as a mom or a dad, you're not allowed to send both copies of a gene to make the baby. You can only send one. They must segregate or separate. That's called the law of segregation, and that happens during a process called meiosis. Law of dominance. If you have two members of a gene pair, one's dominant, one's recessive, the dominant one will overpower the recessive and not allow the recessive one to be expressed. The dominant one gets to be expressed or used. The law of independent assortment relates to the law of segregation. If you have more than one trait in a problem at one time, then those gene pairs segregate independently of each other. In other words, if this gene pair A, separates and one goes to one cell, one goes to the other, that has no impact on the next gene pair, and so on. They independently segregate. So when Mendel was working with his pea plants at the monastery, there were 7 traits that he studied in pea plants. Now, pea plants have a lot more than 7 traits, but these were very easily identifiable as he was growing peas. And we'll talk more in class about why he chose peas as his organism to study genetics. So you notice the way this chart's laid out. He has the trait name like seed color or flower color or pod shape. Then he has the 2 possible outcomes: the dominant one, the dominant allele, and the recessive allele. So for example, let's look at seed color. Seed color is the name of the trait. That's the name of the gene. One possible expression of that gene, or one allele, is the yellow one, and one is the green one. And it turns out that yellow is the one that's dominant. Now, in class, we're going to talk about the rules for writing dominant and recessive alleles and giving them a symbol to abbreviate them by. Okay? So you'll need to be very familiar. There's actually a quiz on this, a 28-point quiz. It'll come up in a few days after we start the unit, maybe 2 days, maybe 3 days. And you have to know everything on this chart. You have to know the trait name and the 2 possible outcomes and which one's dominant. So there's going to be, at first, there's going to be 2 kinds of crosses, genetic crosses, that we're going to be doing. The simplest is the monohybrid crosses. It is so critical that you understand how to do this, because if you don't understand how to do monohybrid crosses, you will not be able to do the other ones that come after this. And I plan on doing 5 types of crosses, 5 types of inheritance. Monohybrid and dihybrid involve the same rules, but we're going to call them two different types. So we're going to talk a lot about odds and chances and percents and ratios. So just want to remind you about different ways you could write that. Like if I flipped a coin, for example, it's got a 50-50 chance of becoming heads or tails. That's 50%, 50%. You could also say it's a 1-to-1 ratio, or a 1 in 2 chance of becoming heads, or a 1 in 2 chance of becoming tails. If I had a normal six-sided dice, I wanted to roll a 4, the probability of that is 1 in 6 because there's only one 4 on that six-sided dice. The probability of drawing a jack out of a standard deck of cards. There's 4 jacks. The deck of cards has 52 cards. That's a ratio of 4 to 52. That will reduce down to 1 to 13. That's like the same as saying 1 over 13 in math. The probability of winning Mega Millions lottery is about 1 in 260 million. So those are what we call high odds. Probability can also be written as a fraction or a percent. So 1 to 2 is also 1/2, and it's also 50%. Past events do not influence future outcomes, but gamblers don't seem to understand that. We'll talk a bit more about that later. Now, I would copy this down as neatly as I possibly could and use it as a guide to when you're doing your first crosses until you get the hang of it. This is the proper way in my class to do a monohybrid cross, and you need to do it this way. Okay? So, but I'll hold your hand and we'll work through these together in class. Okay? That's it. Enjoy your Sunday and your Monday off. See ya.